Hello.
Myount, how are you today?
Good.
Good. Excuse me. Sorry. Oh. Cold.
Yeah.
Yeah. It's turning. The weather's turning. Oh. I'm ready for a break.
Yeah.
You gonna take a little break over?
Yeah.
What are you gonna do? Anything fun?
So I'll visit my friend in Atlanta.
Ooh,
Like,
fun.
yeah. Hopefully less colder than you.
Yes, I hope so.
Yeah.
Enjoy your trip.
Yeah. How about you?
We're gonna be home on actually Thanksgiving Day, but
Mhm.
the day after we're going to DC, Washington DC for a long weekend. We have an exchange student who's here from Italy. Yeah, and she wants to see
Yeah.
Washington
Yeah.
DC.
I've been DC, and what I really liked was the museum was mostly free.
Oh yeah,
Except
most of them are free. That's correct.
except the Deuces,
The what?
Deuces, like Deuces
Can you
Museum,
spell it?
Newnewsis, Deuces.
Usis. I don't know what that is.
Some Deuces Museum.
And the Holocaust Museum. I don't think that's free either.
I think it was in the same.
Was the Holocaust Museum free? I
I
mean, it's a very sad museum, but it's also one of my favorite.
Yeah. Museum, Newsum.
Oh, museum.
Museum.
Like a new.
Museum.
Interesting.
And and the Holocaust is not free as well.
Good morning. How are you, Oksana?
I fall asleep in the bus though.
Oh no, did you miss your stop?
This morning?
No. The app reminds you when you are 2 stops ahead.
Oh,
Like in
okay.
2 stops you have to get down. I'm like, thank God. I don't know what it is with me and transportation.
No, I fall asleep so fast in the car and stuff like that.
Me too. We went to an island once, so the So sheepish. The smaller one, the launch was very like bouncy and wobbly because there are big waves. Like all of my friends were panicking, but they said like I slept like a baby.
Ah, oh my
I
goodness.
don't know. Maybe the rocking of the boat made me even more sleepier. Ready?
Hopefully I expect photo.
Good morning.
Morning.
How's everyone doing?
Good.
Ready for a break?
Yes.
Anyone doing anything fun?
I'm traveling day after tomorrow,
Where are you going?
India.
Oh, with Ashwini?
Ashwini,
Yeah,
yes.
good. Awesome. You don't have any children, right?
No.
No. Oh, that's right. How long you going?
So I'll be back by 1st December.
By what?
December 1st, like I'll be back.
Really? That's a quick trip.
Right.
Yeah. So you're gonna miss class.
Only the 27th maybe, but I'll be available on Zoom. Like if you can
Oh.
have Zoom, I can keep like I'll be online.
Not for this class. Just enjoy your time.
Thank you.
Yeah, sure. Young's going to Atlanta.
Yes.
How about you, Oksana? Anywhere fun?
Well, my friend just had her baby. Like she's the first one to have baby in our
Yeah, your group.
Yeah,
Yeah.
so if all of us can manage, maybe for the weekend we can go to.
Where does she live?
So she recently moved to New Jersey.
Oh, okay, not too bad.
Yeah, she finished her thesis PhD
Yeah.
last semester. So yeah, a lot of big things happening to her.
Awesome. That's awesome.
Yeah.
Yeah, a lot of big things all at once. How about you, Matt? Any?
So I work Thanksgiving morning.
Oh.
Oh.
Pharmacies are still open, so
Okay.
Yeah,
Yes.
right.
I get paid time and a half though.
Oh,
so
that's
it's-
good.
That's
makes
good.
it a little worth it.
Are you? You're not married, right? Are you? Or partner or anything like
No, from far, far away.
Far, far away. Is your family in Pittsburgh?
No, they actually. So my dad retired a few years ago, and so they just moved earlier this year to Colorado.
Oh.
That's nice.
And so you don't go get to. So you're gonna be alone on the.
No. So I actually had a couple friends that just moved back to Pittsburgh. They were living like on the West Coast for a few years.
Oh, okay.
And they don't have family in the area anymore either. So I think we're just really getting
Oh,
rid of
good.
something better, which would be
Good.
nice.
That will be nice.
So what are you doing now?
Yeah, so we, as you know, we have an exchange student from Italy, so she wants to have a traditional Thanksgiving dinner first, so we're gonna do that at home. And we'll have one of, our friends come over, she's a single mom and has two, actually one of her daughters is, very, I guess, best friends with my younger daughter, Molly, they go to school together. And then she has a young, like a toddler, so they'll come over for Thanksgiving. And then on Friday, we're gonna go to DC for 3 days because again, Christina from Italy, she wants to see the nation's capital. So, that's where we are, that's what we're doing.
You have packed weekend.
What's that?
Like you have a busy
Yeah.
vacation. Yeah.
And I have to do work because I just have to. So it's gonna be fun.
Oh yeah, the Friday and the weekend is going to be filled with studying.
Right, yeah. Monday and Tuesday will be definitely like pretty much full-time work for me. And I'm hoping I can take at least Wednesday and Thursday completely off. And then Friday, when we're back in when we're in the hotel room at night, I'll work again. Yeah. Just to get caught up, get ready for Monday.
Is it like the teaching staff or the administrative staff? Research.
Teaching.
Teaching staff.
This semester with teaching has nearly killed me, I'll be honest. It's nearly put me in my grave, I feel. I'm like, ugh. It's a lot.
Yeah, Dr. Wittenderby is thinking like that for her spring. Like she's dreading her
Oh,
spring semester
I
because
know.
she's back to class.
Yeah, most of us have one semester that's like just overwhelming, and mine is usually fall, but spring will be easier, but it won't be completely blank. Hi Melanie, how are you today? Yeah, we were just talking about Thanksgiving plans. Any plans?
Oh, I'm going to Delaware next.
Oh, who lives in Delaware?
I have Filipino friends there.
Great, awesome.
Yeah.
That'll be fun.
Yeah.
Yeah. All right, well, Young, you're up,
Yes.
right?
Yes.
Okay.
Hello.
Hi, Young.
Hi.
So today I'll explain the following lecture from the previous week. So this is more about calcium effector. So today I'll cover the definition of what's calcium effector, and then they have, it depends on their types, they have like a calcium binding, domain or motive, or they are categorized by their different types. Also, I'll explain about the function, some of the functions, and mostly I cover the calcium carmodulin dependent target, and also the rest of the enzyme which is a calcium-dependent enzyme, not regulated by the calmodulin. And the last part of the textbook is something about like a calcium effector regarding on the muscle contraction. And the last, I'll cover one research article regarding cyclosporin A, which is immunosuppressant. So, so what is calcium effector? So, so calcium effector is kind of like a signaling protein which binds at the regulatory side of the protein. So there are certain, I'll explain this a little bit later. So there are certain types. There is active site and then regulatory site. So these calcium effectors binds at the regulatory site, which is a little bit like interesting. And then there are at least 5 types of this calcium binding protein. The first one has EF hand motif, which is shown as here. So I'll explain like this. So let's let's try it. Try to see like this, and then so this part is alpha helix, and this part is alpha helix as well, and then these two parts are connected with the loop structure, and the calcium stays in the kind of like in the middle. Yes, yes. So kind of like our hands.
That can be our new class symbol.
And the other type introduced in this textbook is C2 domain, which is a little bit not like familiar, but as we can see, so this is like just to follow my the pencil. So there are 4 beta strands, and then another 4 beta strain, and then in between these each beta strands are linked with the loop. So let's start from the number 1. So they kind of like a shapes like this, and then when they link to another like subunit, there is a loop, and then kind of like a trebles, like I don't know, it's like
Almost like a figure 8.
yeah. So these are the example, like a synaptotectin or PKC beta 1 or phospholipase C or PLA2. So these are the examples. So so the previous one was I showed just one, the EF hand motif, but it just not just a single one. So mostly they work like at least like a 2. And if we see here and the previous one, for the binding, there are at least 2 calcium needed. And on the other hand, this one, they can hold like at least 3 calcium in between this structure. So the third one is calmodulin dependent or calmodulin independent types. So which is this part is like a kind of like a mixed in the textbook. So some of the ADPase, which is this one of the ADP, calcium ADP in the muscle, the calcium storage organ, which is sarcoplasmic reticulum. This is type of calmodulin independent. However, there is a calmodipine like a dependent ATPase, which is this one. So it in the textbook it's kind of like a mixed, but I try to like a little like say a little bit in detail. So this one is plasma calcium ATPase, which binds with the calmodulin, and the last part is kind of like holding calcium like a buffer. So there are at least like for example like the first one, laticulin or crystalline is kind of like a calcium buffer in the ER or SR, which is endoplasmic reticulum or sarcomeal reticulum. And the last one, the PARP albumin is also like a calcium buffer, and another two is kind of like buffering calcium, but also it it makes like the cytoskeletal structure. And the function of this calcium binding protein is first like buffering calcium, and the second one is pumping out of the cell, which is in the plasma membrane, or storing calcium in a specific organ like EROSR. So which is the example is circuit, and there are calcium sensor as well, which is troponin C, that regarding with the muscle contraction, and the glycovolin is kind of like a urocalcium sensor, and the main part in the slide is more about the signaling regarding like a calcium carmethylene kinase or MLCK, which is in the muscle contraction, and post adenylyl cyclase or phosphodiesterase, and calcineurin or PKC or PLCA, and nitric oxide synthesis. Also, the rest function is calcium calcium binding protein, which functions as a holding the cytoskeletal like actin. So these are the carmethylene structure without the calcium at the first one. So mostly it's kind of like packed inside of the this structure, but when carmethylene bind with the calcium, this there are conformational change, which make this structure kind of like a stretch. So as we can see, there are 2 calcium and another 2 calcium, and these structures, so hydrophobic part is kind of like a packed in the absence of calcium, but after they bind, they kind of like stretch it, and then this hydrophobic part is it will interact with other protein of the hydrophobic part. So in this case was MLCK, and this is the MLCK protein. So as we can see, they are interacting. Yeah. So so basically when they activate, they need calcium, and after they bind with the calcium, they function as this kind of various function. First is protein phosphorylation or dephosphorylation, or calcium transport, or cyclic nucleotide metabolism, yes.
So question, can you go back to the previous slide for a second?
Yes.
So you're saying A is before calcium binds,
Yes.
B, calcium is now bound,
Yes.
and then C is going to be the next slide is showing the list of the different things that
Yes.
the calcium commodulin complex joins with in order to have functions inside.
Yes.
Okay, thank you. Excuse me, sorry.
Yep. So for this part, is the example is when in so for example, like a cyclic ANP can be generated by these 2 enzymes from the ADP, and the rest of them are cytoskeleton and the nitric oxide formation. So these are example of how this chemodulin phosphorylate this phosphorylase kinase and work as a breaking down of the glycogen. So literally they bind, then they kind of like integrate to this phosphorylase kinase. Then, like the name suggests, we can kind of guess it's gonna phosphorylate. So the effector is phosphorylase B. So this enzyme is phosphorylated. Then this enzyme kind of like breaking down the glycogen to its subunit. Not just this function, this chemodulin calcium complex also can integrate into the glycogen synthase kinase, and this kinase also phosphorylate glycogen synthesis A, so that this glycogen synthesis A cannot make the glycogen. So this step is blocking, and this also this glycogen is keep like breaking down. Yeah, so chemodulin works as a like a phosphorylation enzyme with this figure. And so this is an example of calcium chemodulin activated protein kinase. So usually they in the initial stage they are inactivated. How? So like this. Okay, they kind of like packed inside. So which is auto inhibition part in the textbook, and after they bind with the calcium chemodulin complex, then kind of they stretch it out like this.
Oh, I see.
Yeah.
So where's the binding? Oh, it binds to that second domain, the
Yes.
regulatory domain.
Yes.
Okay.
So for this calcium chemodulin kinase has at least 4 types. So the first one is everywhere. So and mostly in the cellular localization is cytosol, and one of the target in the example is this CFTR, which is the probably like a cystic fibrosis transmembrane regulator, and it is phosphorylating this CFTR. And the second type
Wait,
is
before you go to that, so for cystic fibrosis transmembrane regulator (CFTR), the calcium calmodulin kinase 1 phosphorylates that chloride ion channel.
Yes.
And does that then block the function of that channel or activate that channel? Do you know? I don't remember, honestly. You're not sure either. That's fine. That's fine. I just wondered because I don't know if there's an increase in CAM kinase 1 with cystic fibrosis or decrease. That's what I was trying to understand. Because I know they have genetic mutations in that channel. That's what partly leads to cystic fibrosis, at least. So, yeah, I don't know. It's just a thought I had. You don't have to know. I'm just thinking about it. Yeah.
And the second type is mostly expressed in the brain, and that's the major type of this kinase, and as functions such as like it's probably in the neuronal cells, and it is like acts like a learning or like a memory. So yeah. And the type 3 is mostly in the translation part. So this is circulate elongation factor in the protein translation part. So that kinase phosphorylate that elongation factor. And the last one is also in the neuronal cell or mirror germ cell or lymphocyte, and then mostly in the nucleocyte and is phosphorylated the transcriptional factor. For example, like in plant. And other chemotherapy-dependent enzymes are the first one is the plasma membrane calcium ATPase, kind of like maintain the calcium concentration right outside of the cell as major calcium, and then cytosol almost like a very low concentration, and then there are a little bit more, at least 100 nanomolar in the ELSR. So that plasma membrane calcium ATP is kind of like maintain the concentration of the outside of the cell membrane.
Maintains, pumps it out.
Yes,
basically.
yes, basically that pumps it out. And
I'm sorry.
second example is last guanine nucleotide exchange factor. So that is regarding like it's kind of like a guanidine exchange factor. So to activate the LAS, so after GTP bind, then the guanidine nucleotide exchange factor hydrolyze GTP into like GDP, then it activates, right? So that's also chemotherapy independent. And the third one, which we will focus on the research article. So the carcinoline that's activates the NFAT. So if we first look at the structure, so these are carcinoline A subunit and B subunit, and this cyclophilin A is usually that's inside of this two subunit, and it kind of like acts like change. So for example, like isomerization. So from like a cis to trans, the reaction that cyclophilin A is involving, but by like a kind of binding this cyclosporin A in this site, the calcium carmidylene cannot like bind with this pathway, so it blocks the downstream. And so, for example, like calcium carmidylene binds at this subunit of the calcineurin A subunit, then this then this NFAT, this transcriptional factor works, but upon this cyclosporin A at that site, this step is blocking. So the transcriptional factor cannot function at that stage. So in the lymphocyte, T cell lymphocyte, it can activate the cell. So yeah, this much really.
Can't wait. So what's the receptor up with the?
So the first one is ORAI.
Okay, and then so that's an ion channel. It looks like, correct?
So the one that we saw ORI channels for
Hmm.
replenishing the internal stores.
Yes, right. Okay, so then calcium comes in. Okay, good. Then calcium binds to comodulin.
Yes.
Okay, and then that regulates what? That's right.
So So upon this binding, so it also bind with this calcineurin A subunit
Oh, okay.
at this portion.
And does it inhibit it or activate that calcineurin A?
Activate.
Activates it. Okay, got it. All right.
And the rest of there are two more examples. One is adenylyl cyclase and phosphodiesterase. So in their pathway, carnitine calcium is required. And the last example is nitrogen oxide synthesis. So in a cell, we have three types of nitric oxide synthesis. One is derived from the endothelial, and the second one is mostly from, I guess, from the neuronal cells, and the third one is macrophage. And two of them is calcium dependent. The first one is the E nitric oxide synthesis, and the N nitric oxide synthesis is calcium calcium dependent, and the last one is usually in the resting state, but is activating a certain like a portion, and these are the sublocalization, subcellular localization. So, yeah, OG, caveoli, and cytosol, and ER, and caveoli, and fibosome. And the next type is calcium-dependent enzyme, which is not regulated by the chemodulin. And the first example is neuronal calcium sensor. If you remember the first slide, that was like low lactobulin. So that's also calcium sensor. And the second example is caffeine. So this is interesting. Is I think it's phosphatase, and it functions in cell death pathway regarding the neurodegeneration. And the third example is synaptotacmin, which binds with the calcium to release neurotransmitter or hormones, and is in the secretory vehicle or granules. And this example I explained and the next example is calcium calcium buffer is in the calcium buffers is requiring in the cytoskeleton. So this red like a fiber-like structure is actin filament, and there are plus side N, which is hydrolyzed side, and plus side is this subunit is keep adding. So it's then in from this actin filament. So to make complex structure in the cytoskeleton, this alpha-actinin is kind of make cross-linking in this cytoskeleton to hold the structure. And on the other hand, this gersolin is breaking down at some portion, then bind with plus and is capping, and is making it's like a breaking down and making capping. So these are the example. So there is no like a calcium showing here, but yeah, it's also required. And the next example is so this so from this diacylglycerol can be possibly related by this the kinase, which is in next step. Upon this ATP, it makes phosphatidate, and next stage that the kinase is requiring the calcium carbonate. Yeah. And the last part is on the contraction of the muscle cell. So these fibers are actin filament, and then in between there is a myosin filament, and when the cell shift from the relaxation to the contraction to contract the muscle cell, one type of the troponin, which is troponin C, binds with the calcium to contract the muscle cell. So this, and another type is MLCK, which is also phosphorylating myosin light chain. So this enzyme activated upon this high concentration of the calcium, and this from inactivate and activate is chemically dependent, and this enzyme literally phosphorylate this myosin light chain, and make the muscle contraction. Yeah.
So
this,
before you go to your paper,
Yeah.
any questions for Myount?
I have a question in general for calcium channels.
Yes.
Vivek, if you know, you can also answer this. So one of my question was, when does the calcium channel gets desensitized with time? Like we know it happens with some other channels.
Mm-hmm.
Like I was wondering if ion gated channels get desensitized or like cause resistance to any drug.
You mean ligand gated ion?
Ligand gated ion channels? Yeah.
That's a good question, actually. I never really thought about it. I mean, we know G protein coupled receptors do, right? We know other types of receptors do. But I wonder, ion channels. There's a lot. I'm trying to think. This is a question for Dr. Whitander because she knows a lot about receptor recycling and arrestins and all that, beta arrestins and stuff. And I don't know. And I've never really thought about it. That's interesting.
Yeah, because as I was preparing my
Yeah,
presentation,
yeah.
it pops into my mind.
Yeah. And you didn't find anything on it?
No, not really. Like no direct correlation like that, but I do have to look more. I just did a quick Google search.
You know what I think maybe though, maybe not in the same way we think about it, and the only reason I'm thinking of this is because ion concentrations are tightly controlled in the cells, right?
Mhm.
So I think, and they're only going to enter cells if they're going down their concentration gradient, right?
Yes.
Or exit cells if they're going down the concentration gradient. So I'm thinking if there's a lot of ligand for, so let's just say a calcium channel on the membrane and it opens, I think at some point calcium's going to stop going into the cell, right?
Yeah, the
Because
gradient will be balanced inside
right,
and outside.
so maybe that happens faster than the desensitization of the channels. Does that make sense? So it's a desensitization in a way because calcium's no longer coming into the cell, but it's not necessarily a down regulation of the ion channels because it kind of controls itself in that way. Does that make sense? I don't know. Maybe. I don't know. And
No.
And it could be both happening. You know, it could be also down regulation of the channels and this equilibrium being reached between the concentrations inside and outside the cell.
I have to look again. It was just a thing.
Yeah,
that
no,
popped
it's a
into my
good
mind last
question.
night.
Anyone else read anything about that ever? I mean, I don't. Yeah. Any thoughts, Vivek? Since you did the first part of this chapter, anything?
I can just think of like one thing. I don't know. I think they never reach to the level of desensitization because the negative feedback mechanism is so quick. The channels open so briefly,
Yeah.
and once the channel opens, the calcium, low affinity calcium binding sites, like they expose, which are like inside the channel actually.
Mm-hmm.
And when the calcium start running in,
Sorry.
the exposed low affinity calcium channels, they start acting as a regulatory
Yeah, it
feedback
regulates
thing, and
itself
immediately
and then,
closes the channel. So I don't know, even if they reach that level of desensitization, like they're not.
I mean, desensitization
Yeah.
happens with time, and it takes
Right.
chronic
So
exposure
I
of
mean, I'm
something,
guessing
right?
even if the ligand, if it's a ligand-gated ion channel and the ligand is continuously there even after all these self-regulation negative feedback mechanisms are into play, and even after we reach equilibrium with the ion concentrations, I think then maybe there's some kind of desensitization or downregulation, but I don't know. Like just because I think ions, it's so important to the cell to keep it so tightly regulated that there's so many other mechanisms in play that down regulation takes a little bit longer than would be necessary for the cell to act on, right? If they were reliant on just down regulation of the channels, the ion concentrations would quickly go out of whack, right? They would go out of the physiological range.
Yeah.
There must
So-
be something because they are kind of important for all the crucial functions,
Mhm.
cardiac activity, the
Yeah.
neuronal
Well, that's what
cells.
you were saying, though. I think it's because they open so quickly and close
Yeah.
so quickly, and then there's all these negative feedback mechanisms that perhaps that's the mode of desensitization, not the same as what we think of with other types of receptors.
Yeah, because with the G protein, it's complex. It amplifies the signals way more than the
Okay.
ligand gated,
Mhm.
but it says it in the book because it is a complex form of signaling mechanism and has lot of factors like downstream second messengers working on it. So sensitization and desensitization kind of depend upon multiple variables.
Right.
Whereas in ligand gated, it's kind of straightforward.
Right.
So yeah.
I'm just cold. I'm not leaving. I'm just
Right.
sorry, just no. Yeah. No. Yeah.
That was a definitely a good question, I would say.
Yeah, I
So usually receptors get desensitized.
I have to look.
Right. So you
For
would think
myself
that there
as-
would be
Right.
some desensitization
Never
because,
thought of it. Yeah.
but I've never thought of it really like that. Yeah, that's a good question. Well, keep looking and see if you find anything.
Yeah, I
Before your presentation, we can talk about that, and I'll ask around and I'll look too, see if I can find anything.
Yeah.
I'll definitely ask Dr. Wood Underby because she's really into receptor desensitization, that kind of stuff. So, but I never really thought of it till this day. Good question. What do you think, Young? Any thoughts on it?
So I had two thoughts while in this discussion. So as far as I remember, the channel opening, or like the voltage was like the basal level was very low, right?
Right.
That's what I.
You mean inside the cell of
Yes.
calcium? Right.
Yes.
Yes. Okay.
Yes. So it would not like reach the threshold or like just above, not above, and because the basal level is very low, so it might be, I don't know, we may not like really like interested in like because how their reason always show a hyperpolarization, right? Then then
I'm not following. So break it down. So what are you? So you're saying so? Okay. So the inside concentration, inside the cytosol
Yes.
cells, calcium concentration is kept very tightly regulated, right?
yes.
So then
So
what?
so the quantity, like the how we sense the calcium might not be possible, or we may not have enough information because the level is so low.
Oh, I see what you're saying.
And
So if the level's so low, how could we see something that would downregulate even further?
Yeah.
Is that what you're saying?
Yeah.
Yeah, that makes
Yeah.
sense.
That's
Like-
Yeah.
also another thought is so the the GPCR and the channel is different.
Sure.
Both are very macromolecules, but
Mhm.
I guess the channel might have more subunits, and then the structure is very
Complex.
complex, also huge.
Bulky. Yeah.
Yes, and I'm not sure how it can often downregulate or upregulate compared
Right.
to the other
GPC. Yeah, that
other
makes sense too. Yeah.
GPCR.
That's true.
So
The only
This-
thing I could think of is if the entire membrane where the channels are was kind of like taken into the cell, like endocytosed, and then
Okay, so there would be no cell for calcium to gate for a calcium to enter into the cell.
right, or decreased channels, right? Because if it's an area of the membrane where the channels are, and for whatever reason, maybe it's not directly related to downregulation of channels. Maybe something else needs to happen. Something else is close to that channel in the membrane that needs to be downregulated for the cell. So when it happens, it just clips off some of the channels as well. Do you know what I'm saying? Like when we have those clathrin-coated pits
Yeah.
with the beta arrestin and all that stuff, bringing
Yeah.
the receptors in. Maybe there's a channel right next to that receptor that's being taken into the cell. So it's kind of a side effect of the downregulation of other receptors. I don't know. Because it is so big, they are much bigger than the.
And for the endocytosis, probably we focused on very small molecules, so clathrin or caveoli or micropinocytes. They are
Right.
very small. So, so
Yeah,
that channel,
yeah.
those subunits are so big, right?
Yeah, it's a good question.
I will also look for myself as well. Because
Sure,
I didn't
sure.
have the time.
Yeah,
to.
no, that's okay. No, it's something we can all do over break, kind of look for that, because that's an interesting question. Okay, all right. Any other questions for Yon? Who's on to his research paper?
Thank you for very pharmacology.
Yes, it is. Very pharmacological question. Dr. Winterview will be proud when I tell him.
So this paper is explaining the calcium calcineurin regulation that arc the regulation pathway. So which part is interesting in this paper is there is already conventional drug, as name suggests, is working as a cancer treatment. The model, but by adding the cyclosporin, this affects another pathway, which is blocking literally alternative pathway, which caused resistance in this lung cancer. So this paper came across. I found it's interesting. So these are the conventional the pathway of this cryogenic. So this first we have to focus on this MET. So that's receptor tyrosine kinase type of receptor tyrosine kinase bind with the plasma membrane, and it activates this RAS-MEC pathway, and it also affects with STAT or AKT pathway. But so this pathway was probably predominant in the cancer, some specific type of the cancer. Then people developed this hydrotinib to inhibit this receptor tyrosine kinase pathway to make the anti-proliferative this drug. And another concern was yeah. So there were concerns about the recurring the cancer. There is alternative pathway. So yeah. So this calcium affects this calcidurin, and then this pathway affected resistance of this cancer. So that part was the concern.
Wait, stop.
Yes.
Sorry, that little blue is calcium, right?
Yes.
So this
Crizotinib.
crizotinib.
Why
These
you
drug
do
names drive me crazy.
Yeah.
It increases calcium concentration. How does calcium
Reduces
influx?
calcium
How
influx.
did it say, the paper say how it increases calcium influx? Does it activate? I guess it activates calcium channels on the membrane somehow.
There was like a detailed explanation, I guess.
Yeah. And then calcium interacts with calcineurin,
Yep.
which, what's that, KSR2, which activates, okay, I see, okay. So it's kind of like the drug inhibits the RTK, the MET, the receptor tyrosine kinase, right?
Yes.
But
It also
it also activates, interesting.
yeah.
So it's kind of, it's kind of fighting against itself, looks like,
Yeah.
right? Yeah, okay, all right.
Then this paper proposed modulating this calcineurin using this cyclosporine. This pathway will be affected. So they want to show how this cancer cell can be sensitized on this cyclosporine.
Okay. Yeah,
Did they say how resistance develops in
So I think
So-
yeah. So these are the resistance.
that's the pathway.
Yeah, yeah.
that's the pathway they're
Okay.
saying resistance.
So do you know if this side of the pathway, did they know that one first, and then only when they saw that patients were developing resistance did they find that this pathway is actually involved? Do you know the timeline? I'm just curious about the
That's a
timeline,
good point. That's a good point. Yeah.
because usually like a drug is approved with this particular indication and this is the mechanism, but then later on they find that it acts on another pathway and other things are happening. So I'm just
Logically,
curious
this should
about
be
which
first.
one.
Like, the
Yeah.
right side should be first.
Yeah.
It got approved
Yeah.
using that.
Yeah.
Yeah.
Yes, and
So
then they
this-
But
find
then they
this.
found that this is also
Yeah,
happening.
right, right.
So, like you hypothesized, this pathway would be the first. Then
Yeah.
they found another. Yeah.
Okay.
Maybe they saw like the drug did not reach its full potential because of
Because
like,
another
shifting,
thing's
and then
happening.
they started
Yeah.
using combinations with
Yeah.
cyclosporine.
Right.
So they used two types of cancer, which is pancreas cancer, which is left cell, and non-small cell lung cancer, which is the right cell, and the crizotinib is blocking
It's hard to
cell
see the symbols from back here, I'm sorry, I'm just
so-
trying to. Oh, yeah, okay, so the top
so the top
is this,
is
the drug alone.
yes, and this is in the presence of cyclosporine. So it inhibits proliferation and yes,
It's not super effective, but it is significant.
Yeah. also so it showed in pancreas cell as well as this lung cancer cell across various concentration here.
Yeah.
And they also tested the number of the colony using this colony formation, as in when they quantitated.
So stop for a minute,
Yes.
Young. I want you to take a deep breath,
Yes.
and I want you to walk us through this slide so we can understand what's going on here. This will be a
Yes.
good practice for you. Go
Yeah,
ahead.
exactly. Yeah. So, so this left panel shows the colony formation assay result, and the top panel is the pancreas cell
Good.
result, and the lower panel shows the lung cancer cell
Okay.
result. And these are vehicle control and cyclosporine treated group and drug treated group and combined group.
Okay. And what are the colonies? The more blue color means more colonies. Is that the
Yes.
idea? Okay.
Yes. So this blue dot means colony. So so
Okay.
yeah. If we
So what is that showing then?
So it's showing compared to the vehicle control, this cryogenic and the combination is inhibiting this colony formation and
Okay.
is enhancing its inhibition effect in the presence of the cyclosporine.
Right. And then what are those graphs?
Yeah.
Are they just Are they just confirming it? What are they showing us?
Yeah. So this right panel is the quantitation of the colony numbers.
Right.
So compare this. So these are vehicle control and cyclosporine treated group and drug treated group and the combination group. So
Good.
yeah.
And what's C? Is C the lung cancer or the pancreas cancer?
Yeah. So this is the pancreas cancer and this is the lung cancer. So
Very good.
yeah, both of them they show the inhibition of the colony formation in the drug treated group, and it was enhanced by the cyclosporine at both. the
Interesting
cells.
that it doesn't seem like they did the statistics, or did they do?
Yeah, they did.
Did they? Yeah, but I mean between control and the treated groups.
Yeah.
Do you know if they did those statistics or not? Do you remember? I mean, it looks significant,
Yeah.
but I just did. You can't always go by how it looks.
One good thing is cyclosporine alone is not that effective, and when you see the combination, it's
Yeah,
Yeah.
right. But I'm wondering, like, the the drug alone, CRIS, whatever that
Right.
long name is, and then the combination, are they significantly different from the control? Do you see what I'm asking?
Yes.
Mhm.
So
basically control versus combination.
Right.
assume,
I mean, it looks like
They
it.
do,
yeah.
they do look.
It does look significant. However, you can't. I mean, I personally would like to see their.
Maybe you can calculate it, young.
Yeah, in your free time, just go back. They'd have to give us all the data points in order to do that. So, yeah. But it looks significant. Okay,
Yes.
all right. I just wondered if they did it or not.
Yeah.
It doesn't look like they did it, but maybe they mentioned it in the text. I don't know. Okay. Okay, same thing here. Walk us through.
Yes.
Okay.
So this is let's see. So left top panel shows the calcium level in the cell
Mhm.
using this dye, which is fluorescence probe to plot the calcium. So the left top panel is the pancreas cell and lung cancer cell, and so as this graph shows, this drug is increasing the intracellular the calcium level using this probe in both cells across the time, and after after the maximum time, the level was decreased.
The drug concentration is different though. Like they use 2 micromolar
Mhm.
and then 4 micromolar.
Yeah.
So A is looking at what?
So intracellular calcium level,
Okay. With increasing, with time.
yeah, with time.
We can see it increases in both cancer cells.
Yeah.
And then B is showing what?
Is
Same thing, just a different assay or?
so B is showing the dose.
Oh, dose,
Yeah.
okay. So they chose 2 micromolar
Based on
the maximum
Yeah.
response. Okay, all right, okay. They could have chose 1, 2, but okay, got it. Okay, so basically in both cell types, Cristoninib or whatever it is increasing calcium concentration, right? Okay. And they use that, what is that, some kind of fluorescent
Yeah,
assay for
fluor-
calcium. Yeah, okay. And then what's the right-hand side showing us?
So so the white top panel in both the cells, so they use the toxicity, which is
What is it, I'm sorry?
toxicity. You may say
I never heard of it. Can you spell
that.
it?
So THAP.
Oh,
Yes.
okay, I got it, Thapsicargan. I've heard of that, okay, yeah.
I think
So that,
it's
is that
also-
known to activate ERK1/2 then? That's like a positive control thing, or what is that for? Why are they putting that there?
So so I think it's blocking the release of calcium from the calcium storage.
It's blocking or activating? Because down there you have Thapsicargan increases intracellular
I think
calcium.
it's
So it increases,
activate, yeah.
activates, okay, all right.
Activate, yeah. I remember that might be like something like a stressor. In the
So when you increase calcium with Thapsicargan, what's happening there then?
so the ARC was activated. As we can see, the phosphorylation status was increased compared to the phosphorylated group, and
In both cell types?
yes, in the both cell types. And when they treated this drug, also increases phosphorylation of the ARC. On these 2 panel, but however they kill the calcium using this reagent EGTA. So this is the negative control.
Got it. Yep.
So yes.
So
this-
the EGTA then, which is the calcium chelator or buffer, like you said there,
yes,
that then decreases the ERK1/2 activation by this drug
yeah.
in
So
both cell lines. So a couple of things, I just not for you specifically, but for the class for this paper. What are some of the things you see here that maybe they should have discussed or did, unless they do it next? Can I see the next? What's the next figure?
also that's the trend is same and they're just a different cell. So this
Right.
is vectors and this is
Right, but look, so there's a couple things that I have questions about
Yeah,
here.
yeah, yeah.
What do the students see here? What do all of you see here? My first question, okay, I'm just gonna say it, is why is there a doublet for ERK1/2 in D and not in, at least I can't see one in C. So I'm not sure, I don't know what's happening unless it looks like to me ERK1 is the higher ERK, it's 42 kilodaltons, ERK2 is 44, right? I think, or is it switched? It might be switched. Anyway, the top band, the 42 kilodalton band, is pretty much absent with Fapsigargan, right? Looks like. I don't see much of it. Right. So it suggests to me that maybe something other than calcium is also involved in the bottom panel, right? Because it looks like the top panel, they're only looking at one of the ERK isoforms, either ERK1 or ERK2, because I'm blanking right now which one's 1 and which one's 2. I think 1 is 42 and 2 is 44, but it might be flipped. But either way, they're only measuring the 44 kilodalton isoform of ERK there, right? Whereas below, it looks like you were capturing both isoforms, ERK1 and ERK2. So that's the first thing I noticed. So maybe it's not just calcium, or maybe it is, I don't know. You know what I'm saying? Like, I don't know what that, I'd have to think about it a little bit more what that means. And then the second thing is, look at ERK1-2 total levels in the SPCA1 cells in the top panel. It's definitely decreasing the total. So Dr. Wood-Enderby especially would say they should semi-quantify this, right? And show ERK1-2 total over GAP-DH, phospho ERK1-2 over GAP-DH, and then phospho ERK1-2 normalized to total ERK1-2. To be sure, you know, just to show what kind of things are being regulated here. Yeah. That's my thoughts.
Yeah.
But but overall, it does support their hypothesis that the CRIS drug is increasing calcium concentrations there, and that is then leading to activation of ERK1-2. Yeah. Do you see what I'm saying, everyone? Yeah. Okay. Yeah, good.
And they xenografted these cells in the mouse, and they measured the tumor volume and the tumor weight. So first, the top left panel shows the effect of combination with the cyclosporine among these groups. So the first group is the vehicle control, and in the presence of this drug, it shows the tumor decrease. Also, it's enhancing in the presence of this cyclosporine A. Like they did some better statistics here. And
So wait, so what's the top left? Tumor weight,
yes,
and that is what?
cyclosporine A.
Which, okay, and then, oh, I see, okay. And then on the right
It's
hand side,
treat the PD mechanism.
oh, plus the MEK1-2 inhibitor. Which is interesting because on the diagram you showed, it looked like calcium activated ERK1-2 below the level, like almost directly. It didn't activate MEK1-2, right? Can you go back to that?
So
Right?
So
I can't see that.
The bigger one.
Yeah, go previous.
So
So is PD, what is PD 90?
maybe
Is that the,
No, go to one of your first, oh yeah, this is it. Yeah, keep going. Yeah, there. Oh, yeah, so where does PD work? Where does PD inhibit? PD inhibits the upstream MEK1-2, right? Okay.
okay.
So they're showing, but in this figure, it looks like the crizotinib, I'm just going to go with Criz. The Criz increases calcium, activates calcineurin, then this KSR2-1, which then activates ERK1-2 downstream of MEK1-2. So it doesn't make sense to me that the PD would
Be able to
be able to inhibit that effect. Do you see what I'm saying? Because you're blocking upstream. So you still would see the calcium activation of ERK1/2 downstream. So I'm kind of surprised that the two
So
had.
the down is MEC1/2 like essential for activation. Because that
Not according to this diagram.
so like if MEC1/2 is not there in the environment, can ERK1/2 still be there, like still be present in the absence of ERK1/2?
Yes.
Okay. Yeah, that it makes sense.
But I'm saying, do you see what I'm saying with the figure you just showed us? So remember this diagram in your head, okay? Everyone got it?
Okay.
Kind of, you know, the idea. Not all the, but you. Here, I'll draw what I'm saying. This is what I'm saying. I actually have markers with me because I've learned over time none of these classrooms have markers in them. This is what I'm saying. I'm saying what they showed is, okay, so up here is the RTK, right? In the membrane, you get, I think it was RAS or RAF, whatever, MEC1/2, ERK1/2, right? And then over here, they say the CRIS drug inhibits this, right? Okay, fine. But then they're saying, on the other hand, the CRIS drug over here increases calcium, and then it's like cyclosporin, right? Oh no, calcineurin. Which then activates ERK1/2, right? And there's something else in there, I know. Okay, right? So the first one, B, they have the CRIS plus the cyclosporin A, and so CSA inhibits this, right? Are we right so far?
Yes.
Am I correct? Okay, so they gave this drug, which is inhibiting up here, and this drug, which is inhibiting here. So that makes sense that they cooperate, right? But now they're adding, I have other colors, but just for, now they're adding the PD, which is up here, an inhibitor of MEC1/2. And they're saying when you add that plus this, you're getting cooperation, but this is acting downstream of the PD, so I'm not sure how that's working. You see what I'm saying? Unless they should have drawn this.
Right.
Do you know what I mean? Is
They
that,
they also did the Western blotting in the next.
okay, yeah, let's see what happens with the Western blots. So what happens here?